Hi everybody, I'm Firas Khatib, and today I'm going to talk about the protein folding video game Foldit. Now I always used to start with the question, why proteins? How come they get their own video game? I would then explain with images of HIV, Ebola, or Zika, but now I can just show you this. We've all seen the COVID-19 virus, and what it looks like, it is made up of a bunch of different proteins, and they're very, very small. In fact, they're so small, we're actually going to get a proper sense of scale by zooming in here. So we see a sesame seed, we'll zoom into a grain of salt, an amoeba, zoom in even further, human egg, start seeing a skin cell, red blood cell, and then we start getting to nasty things like E. coli. You don't want that. And measles, the measles virus, made up of lots of different proteins. Influenza, that's the flu. HIV, made up of lots of different proteins. Hepatitis as well. And hemoglobin, for example, is a protein. So that's how small we have to go. In fact, proteins are so small, we can't see them with a microscope. And if we zoom in even further, we see a water molecule and a carbon atom. So proteins don't just make up nasty viruses. Proteins are also in charge of a bunch of functions that are fundamental to life. And looking at this tiny subset here of proteins, you can see that they all look very different from one another. And that's because the structure of a protein determines its function, what that protein actually does. So for example, insulin here has a very different shape than hemoglobin because they perform very different functions in your body. So if we want to understand how a particular protein works, what its function is, we need to find out its structure, what shape the protein folds up as. We also know that each unique protein sequence folds into a unique structure. We refer to that as the protein's native structure, the native fold of the protein. So we need to know the structure of a protein if we want to understand exactly how it works, but this is super hard to do. There are experimental ways of solving the structure of a protein, but they're very time consuming, they're very expensive, and they don't always work. In fact, the gap between the number of known protein sequences and the number of solved protein structures, well, that gap is growing every year. We have a bunch of genomes that are being solved, so we have a lot of sequences that are being solved, but we actually have no idea what those corresponding protein structures look like. So this was the aspect that drew me into the field of protein structure prediction, which is trying to use computers to bridge this gap. And there are many different algorithms to tackle this problem. One of these is the Rosetta algorithm, which was developed at the University of Washington in Seattle to predict how a protein folds up based just on that amino acid sequence. So what Rosetta does is it tries to cheat as much as possible by stealing as many fragments from solved structures to try to then predict the structure of unknown proteins. And so you can see the simulation running here, and you can imagine how these calculations are going to be run on a lot of computers. In fact, even using clusters of supercomputers wasn't enough to solve this problem. We needed more computational power, and you can see if the simulation goes well, it matches the correct answer here, which is in white. So we needed more power, so we turned to something called distributed computing. Now, there are many different distributed computing projects. When I was in college, SETI at home was a big one, where we would go to class and we'd leave our computers in our dorms because we didn't have laptops back then, and they'd be searching for extraterrestrial life while we were learning. I don't think anything ever came out of it, but maybe I just didn't get that email. Who knows? So Folding at Home, for example, is a protein folding distributed computing project at Stanford, and the Rosetta algorithm has its own distributed computing project as well, which comes with the Rosetta at Home screensaver, which you can see is magnificent here on the left. And so there are over 1.3 million users of Rosetta at Home. But even all that computational power, where we're using people's computers at their homes, is not enough to solve this problem. Why? Well, proteins have so many different possible conformations that they can fold into that even with every supercomputer on Earth, it would be impossible to sample every single conformation for a single protein. Proteins just have too many different degrees of freedom. I'm showing you here all the little different degrees of freedom. This is a protein segment with just two amino acids, and proteins have thousands of these, right? So you can get the idea of all the different ways that it could fold up. But what would be great is if we could guide these computations somehow. And I've intentionally left this ugly screensaver up So you would stare at it just as a lot of our Rosetta at Home users used to do. And what happened is some of them contacted us and said, you know what, your algorithm's making some mistake. It's moving the helix this way. It should be moving the protein this way. You need to let us intervene. And so that's actually how Foldit got started. Foldit is a graphical user interface representation of the Rosetta algorithm, where proteins can be manipulated with the Rosetta energy shown in real time at the top. Now, you'll notice that energy in biochemistry, you try to get the lowest possible energy, but gamers do not like optimizing for negative numbers. So the very first thing we learned was that we had to change it to a positive score, trying to get the highest score, obviously, in a video game. So that was lesson number one. Foldit players can form teams across the world and share solutions and strategies with others. So who plays Foldit? Might not be surprising to hear that most Foldit players are men and many are students, but 50% of our players are from the US and the UK, but the other half are not, which is really exciting. And Foldit has been translated into many different languages by the players themselves. Here's a more recent survey from 2019, kind of a similar demographics. But we wanted to know who really plays Foldit. Is it just protein experts that study proteins all day and work in a lab and then they go home and play a protein folding video game? Didn't really think that was the case, but we asked our top players now, how much prior biochemistry experience do you have? And the majority of them said, oh, I haven't taken chemistry since high school, or I don't have any biochemistry experience. And so that's very exciting, because suddenly you're getting a very different angle on a problem where we've had a ton of experts studying this problem for a very, very long time. And now you're getting maybe people looking at it from a different direction. And this is a more recent survey from 2019, where you can see that most players learn all about this protein stuff by actually playing the game, right? But this puts us as game developers in a dilemma, because nobody wants to read a biochemistry textbook while playing a video game. In fact, you're probably already thinking this was way more chemistry than I wanted to hear all year. Well, so because most players do not have a biochemistry background, we add these visualizations in the game. So you might not know anything about proteins, but if I show you a game with a spiky red ball, you know that's bad, right? And so that represents a clash, and it just means you need to pull these two parts of the protein apart because they're currently clashing, right? Filling bubbling voids and empty cavities is another thing we represent with these pulsating spheres. Those are bad. Proteins like to be nice and compact. They don't like to be all open and have big cavities. You want to hide exposed hydrophobics. So we show these little greasy yellow spheres kind of moving around. They're not as bad as clashes and voids, but you also want to make sure they're not everywhere. And lastly, hydrogen bonds are what hold proteins together, and so we show them with these nice little blue lines. And so these are all important game design concepts, right? Because you can make your game super accurate and very biologically realistic, but it's not going to do you any good if non-experts can't understand it enough to be able to contribute to it. But it's a fine line, right? You don't want to hide too much of the science either. I think initially one of the concepts when Foldit got started was to replace these amino acid side chains here with different fruits and different shapes, just so that it was really, really more like Candy Crush. But you lose a lot of the science that, this one is a ring. It's kind of important. So this is normally where I would ask for a volunteer to play the Foldit tutorials in front of everybody and really struggle because this is a hard game. But hopefully some of you already tackled it in the pre-conference Foldit challenge. I'll just briefly show you how it is if you haven't. There's a bunch of different levels that unlock as you progress through the game, and they teach you, again, not all the biochemistry that you need to know, but rather the tools you need to know to be able to contribute to the Foldit project and contribute to the science. So this one is very simple, early one where you have to remove clashes by just splitting, and then you get the fireworks and the sound and all that stuff, and all this positive feedback. So the problem Foldit players are trying to solve is, given a protein sequence, what structure does it fold into? What does the native three-dimensional fold actually look like? And so the first thing we wanted to know is how Foldit players compare to our automated computational methods, the Rosetta algorithm, for example. It quickly became apparent to us, though, that in order to test this, we'd need some blind cases where the native solved structures were not publicly available. Now, what do I mean by this? Well, we previously would just take a solved protein, and then we would intentionally unfold it so it looks all messed up, right? Proteins don't look like this. I mentioned this. They like to be nice and compact. And we give this to the players as the starting puzzle, and we would get amazing results back. And we were so excited. We're like, oh my goodness, these players have solved the protein folding problem. And it turns out they're just very clever. And they just went to the protein data bank, and they just looked up the solved structure and then had it on one screen and would fold this into the correct shape. And we were very sad. So we ran an experiment on 10 blind structures that were not yet released because they were about to be experimentally solved. So there'd be no shenanigans. The structures weren't even available yet, right? And so we ran a comparison against the Rosetta algorithm. So this is just how Foldit did against Rosetta. A lower number is better. If it was a 0, that'd be a perfect match to the correct answer. And so you can see that for 5 of these cases, the Foldit players did better. For 3 of them, they're basically did about the same as Rosetta. And for 2 cases, they did worse. But these numbers are all pretty high here in this case. So those might have been just really, really hard ones. I do want to just show in more detail the case where Foldit players significantly outperformed Rosetta, because it highlights one of the advantages that humans have over computers. So we're going to look at how one player took this incorrectly exposed blue tail. So it's supposed to be buried in the protein, and the starting model had it just sticking out like this. And this is the player's score at the beginning. And we're just going to look over time how their structure and score changed. So the first thing this player did is they started to unravel the protein, which made their score get worse, right? Suddenly there's going to be a lot more voids, and it's not as compact. And then the player did a crazy thing and made it even worse. This is definitely not protein-like. As I said, proteins like to be nice and compact. This is definitely not like any of the proteins that I showed you before. And their score just got horrendous. Now, why would they do this? Well, these were the steps that they had to do in order to get that blue exposed tail here into the middle, where suddenly their score got better. Still much worse than the start, but eventually better than their starting structure score, and very, very close to the correct answer. Now I'm going to show you a crazy plot where we can follow the player's trajectory. Don't freak out. There's a lot of dots here. Every green dot represents a different player's Foldit solution. And the further anything is to the left, the closer it is to the right answer. And the lower it is, the higher the Foldit score is, the better, the lower the energy. So if we just follow the starting model, which was this black dot right here, and we see this player's trajectory over 2 hours, they went in the completely opposite direction, right? Away from the right answer, horrendous scores, but those were the steps that they had to do to get to this really, really close to the correct answer area that they ended up being. So this is the advantage that humans have over computers, because the computer algorithm here is shown in yellow, the Rosetta algorithm, and it started with the same model, and it generated all these yellow points. And probably the closest yellow point is right here. I don't know if you can even see it, but the computer gave up there. Now, why didn't it keep going? We could have easily encoded the algorithm to say, hey, don't give up, right? If you're at this spot, don't give up. But what does that mean? That means that for this spot, we'd have to tell the computer, don't give up. For this spot, we'd have to tell it. And then we get back to the same problem that I talked about earlier, in that it's too computationally complex to try out every single possible solution. So it ended up being that the algorithm did not get that close, whereas the player made the green solution here, which you can see is very, very close to the blue, which was the correct native fold, and the starting structure here was in red. So anyway, these were very exciting kind of proof of concept results for us. But what we really wanted to do was apply Foldit to a real world problem. And the Mason-Pfizer monkey virus retroviral protease is a protein that causes simian AIDS in rhesus monkeys. And experimentalists had worked for over a decade to solve the structure of this protein and had been unable to. And there was this model here, which doesn't really look like one model. In fact, it's 10 different models. And it didn't work for them. Using this model didn't work. And I'm going to show you a different seizure version of it, where you can see all the different versions of this model. And Rosetta wasn't able to solve this. All the state-of-the-art methods were not able to solve the structure of this protein. So it was kind of a Hail Mary. We're like, hey, let's do a last-ditch effort and see how the players can do. We normally post a puzzle for a week. We gave this one to them for 3 weeks over winter break and see how they did. So this was, we gave them all 10 of those models that you just saw. So this was model 1 that a player, SP Vincent, decided to work on. And after 5 days, this was SP Vincent's 2,771st model for this particular puzzle. That's the model here in yellow. And already, this yellow model that SP Vincent made is closer to the native structure than the starting red model. See, the yellow here is closer to the blue than to the red. Exciting, but if we look at it from a different angle, you can see that these core side chains that I'm showing here are quite a bit off, right? Even the yellow and the blue is not perfect, right? So SP Vincent, though, had a teammate named Grabhorn who was able to improve it. Now, SP Vincent lives in Canada, Grabhorn lives in the US, but they were able to share these solutions across Foldit. And now if we look at the core of the protein, those side chains are really, really spot on, the one that Grabhorn worked on. But this loop here on the top left is still wrong. Luckily, Mimi, she lives all the way in England, was able to tuck that loop in. And using Mimi's Foldit solution here in green, crystallographers were actually able to solve this structure. The green model fit their data well enough that they're actually able to solve the actual structure of the protein, this one in blue here, and that's in fact the only way I'm able to show you this blue model because we obviously didn't know it at the time. And if you look how different it is to the starting model, it was a really, really exciting result. It's very different from the first model that we gave them. But for some problems, you need people to think outside the box, right? Foldit players have no blinders on, whereas a protein expert could say, oh, well, this would never fold, so I'm not even going to waste my time trying that. So this was very exciting news to all of us, yet when we asked the 3 Foldit players if they'd like to be co-authors on our journal publication, they respectfully declined, saying it was a team effort, it wasn't just the 3 of us, and so we want our team name, the Contenders, the name of their Foldit team, to be co-authors on the paper. So we published our paper with Foldit Contenders Group as co-authors. And now I'm going to have the players tell the story themselves. Right here.
My mother died from a brain tumor. It really blew me away. So I reassessed my life and thought to myself, okay, I'm going to research cancer. And I think the Foldit advertisement was, you can win a Nobel Prize. And I thought, oh, what's this about?
Foldit is a computer program that allows people with no scientific knowledge to contribute to a scientific experiment by discovering the shape of proteins.
Quick example, you take like the flute, right? You can think of that protein has a sort of shape. What we're trying to design is another protein that would be, let's say, a key that would fit right in the lock, and then it would basically inhibit that protein from working.
The idea was then to basically make a game about protein folding, because it's the same reason that, do you know, like CAPTCHAs? When you want to sign up for like an email account, they show you some text and you have to type it in. The reason that you do that is to prove that you're a person, because people are so much better than computers at processing and recognizing visual information and patterns.
The scientists had been working on the Amazing Pfizer monkey virus for something like 15 years without finding a good solution. Knowing the structure was important to combat the AIDS epidemic, and so they just gave it to us as a normal puzzle, and we had about 3 weeks to work on it. I looked at the shape and I wanted to get one root to tuck in, but I couldn't get it to fit. Then I had to go with a solution that a couple of members of my team, the contenders, had worked on. And this time it worked, so it was really a team effort. I didn't think much about it until a few months later when they called us up and told us the solution we had submitted had enabled them to solve the structure. They were very happy about the breakthrough, both because of finding the answer and because of proving that an application like Foldit can produce results. Then the paper was released, and suddenly we were all over the international news.
When we were writing the paper about that, we actually contacted the players and we asked them if they wanted to be authors, like individually.
Like we contacted Mimi and we contacted the other
do you want to be authored? And they said no, they would rather have their group be the author, the contenders group, which they're all members of.
It's a step along the way to solving AIDS, and that's probably the thing I'm most proud of. Humans will always see faces in clouds. I don't think you could teach a computer to do that. And that's maybe why the scientists haven't achieved finding what the shape was in all these years that they've been looking at it.
We've given our little input so that people can use it for AIDS-related disease. So Foldit is a game of life. Just have a look at yourself. You know, like, you're built up with all these proteins and you're a beautiful person. And I hope one day that all these beautiful things that I can see and the way that I can fold will be able to help someone. It's better than any other game because I can change the world.
So, for the Foldit community, this was a very exciting moment that citizen scientists could contribute to the protein folding problem. Then, in 2020, DeepMind's AlphaFold program came out of nowhere and revolutionized the field. Now, I don't know if you heard about this, but in our community, this was huge. DeepMind essentially solved the protein folding problem, which had been the holy grail in biochemistry. Now, why am I interrupting a talk on video game playing citizen scientists with what seems like a Google ad? Well, it turns out that DeepMind was inspired by Foldit. In 2009, came on their radar and made them think that maybe they could try to mimic that intuitive capability that our gamers were demonstrating. In fact, this was even in his keynote talk at the scientific meeting where these results were announced, where he noted that Foldit players reminded their team of Go players, which DeepMind had previously tackled with AlphaGo. They also did chess before that. Now, while this made the Foldit team and players feel very proud, obviously, many in our field began to sweat. Are we done? Is there no more science to work on if Google can successfully predict the structure of a protein just from its sequence? One reason we're excited to keep folding is because of protein design, which is the opposite problem of structure prediction, and related to the very first protein that I showed you. The COVID-19 structure was solved very quickly after scientists figured out its sequence. That's how we can see it here, and that's how we can fight it. Now, we can't design any drugs or vaccines against COVID-19 if we don't know what it looks like, right? But if we want to design new proteins to fight COVID, ones that don't already exist in nature, then we had to create new proteins. So, let's say the shape of this protein on the right here can block COVID-19. That'd be great. But let's just say this shape here could actually block it. Okay, wonderful. Now, how do we get such a protein to fold into this very shape? What is the sequence that we would put on it that would get it to fold that way? So that is the protein design problem. You already have the folder shape that you need, but you need to now find the protein sequence that will give you that design. So given a general function or structure that you want a protein to have, what sequence is going to produce that one? So the idea is to create new proteins that don't exist in nature. Now, this is not something that the current DeepMind methods can do because their machine learning techniques look at natural proteins, not ones that don't exist yet. So when we started protein design to Foldit back in the day, we created a trophy for the very first player design that we ever tested in our lab. And even though that lab test didn't work out the way we hoped, this was over 10 years ago, we were still very excited about this and sent the trophy to that Foldit player that created the design, Boots McGraw. Here's a picture of Boots at home with his trophy, and his boots, of course. And I love Boots' quote for many different reasons. I'm going to read it real quick. He says, "I'll bring it to the office and leave it on my desk, so when my coworkers once again ask why I don't want to play Farmville with them, I will show them the model and tell them that I'm busy curing cancer, AIDS, and the common cold." Now, obviously, this is an old quote because, Farmville. But it really highlights a lot of motivation for many of our players. Some of our players have been playing Foldit since it started in 2008. That is not your typical gamer, right? Who, as much as you might love a game, will move on to the next one, right? But this is a game with a purpose. Boots' quote also points out how many people play Foldit at work, which is also awesome. We've noticed the number of players drop on weekends and holidays. So just like with structure prediction in Foldit, the first thing you'll encounter for protein design is a series of tutorial levels, where at each position in the Foldit sequence, you're able to decide which amino acid to pick. Now, this isn't easy because there are 20 different amino acids to pick from at every single position. So computationally, that means that for, let's say, 100 residue protein, we'd have 20 to the 100 possibilities for the sequence alone, and that's without even folding the chain, just looking at the sequence. So designing a new protein that folds up into a predicted structure you created is super tough. In fact, Foldit turned 15 years ago this past May, yet we struggled with protein design until a breakthrough a few years ago. Finally, our amazing Foldit players were able to create brand new proteins that don't exist in nature, and more importantly, we were then able to solve them, and they looked almost identical to the player predictions. You can see the superpositions here between the ones that are designed are the rainbow colors, and the ones that were solved experimentally are the gray ones here. Very, very, very similar. My favorite part of the story, though, is that those players who actually created those were co-authors on our Nature paper. Their actual names, not their Foldit usernames. That for me was the coolest part. So our challenge during the pandemic was to use Foldit players' skills to design proteins against COVID-19. We started by launching 2 challenges in Foldit, trying different strategies to combat COVID-19. First effort was to design an antiviral protein. By now, we all know the image from the CDC by heart, with the coronavirus spike proteins in red, all of these ones, right, that form a corona around the virus. The idea is to design a new protein that'll bind to this red spike protein, which is exactly what we did in Foldit. We gave the players the actual spike protein in the game, which they cannot change, obviously, as we can't modify that. But then we let players design proteins to bind it. So basically, we just kind of chopped off here the very tip of the spike protein and just gave them this part, which you can see here. And that protein is fixed. That's why it's all grayed out. But the players here can add, remove, mutate, change their sequences and fold in any way they want, anything that'll bind to the spike protein in the best way. Our second strategy against COVID-19 was to design an anti-inflammatory. The term cytokine storm was mentioned during the pandemic, which happens when the immune system releases more and more cytokines to defend itself. Those are shown here as white dots. But instead, this causes the immune system to go into overdrive, and it starts attacking human organs. The approach we took with Foldit was to try to block these cytokine signals and this overstimulation of the immune system. From a Foldit point of view, this is a similar problem, right? We just need to design these green dots here to block the white cytokines. So what makes this problem so difficult? Well, the first problem is that whatever you design as a player needs to have a ton of small interactions with the target, right? So if this is the spike protein down here, and this is your design, there need to be a ton of interactions between the two, which makes sense, right? But here's the catch. Whatever you design has to be able to fold on its own before it can go and block the virus and create all these interactions. So it has to be something that's stable enough to exist on its own. And then once it gets to the spike protein, it would be able to have all those interactions. So those are two very different things that are often at odds with one another, making the problem very, very difficult. So we tried a bunch of different approaches to tackle these puzzles. Here are two examples. The first one was just starting from an existing protein fragment on the left here. So this is a part of a protein that exists already in nature, and then letting the players just add more to the ends and try to fit something that would block here. And the other method was just giving the players a completely extended chain to fold it up from scratch, really coming up with whatever they could, again, trying to think outside the box, as opposed to trying to use kind of techniques that experts have already tried, right? Now, you might be shocked to hear that Foldit players did not solve COVID. But the idea is to prepare them for the next nasty protein, whether it's COVID-25 or Super Ebola 28, whatever it is. It was also a venue for many people to contribute to fighting COVID during the lockdown. When many of us felt locked up and hopeless, this was a productive place to focus some time and energy. In terms of new users, the second biggest month in Foldit history was when we had our coronavirus puzzles come out. And this was the monkey virus one. After the paper had been released, and as Mimi explained, it was all over the internet and all that. And in terms of the protein design puzzles, which I mentioned came much later, after the monkey virus example, the COVID puzzles were definitely the biggest amount of participation that we've had. But it's not just COVID. Our players focus on many different things. They've tackled influenza by attempting to design flu inhibitors. So this is a part of hemagglutinin, which is this is a somewhat conserved section of the flu, because as we all know, the flu likes to mutate all the time, which makes it very hard to design something to block. But this region here, a little less mutations there. So they're targeting that region. For our 1000th ever Foldit puzzle, which was in 2014, we posted a particularly challenging Ebola design puzzles for player to create virus blocking designs. Again, spoiler alert, Foldit players have not cured Ebola or the flu. But we really think of it as another tool in our arsenal to combat these diseases and many others, right? We have traditional methods. This is definitely not a traditional method, but even if it only helps rarely, it still would be a big win. And Foldit is not just for gamers. Being able to redesign a protein in real time makes Foldit a very useful protein manipulation tool for scientists as well. In fact, using Foldit, undergrad students who were participating in the International Synthetic Biology competition, which is called iGEM, were able to generate an enzyme with a potential as a therapeutic for celiac disease. Now, this was over 10 years ago, but they've recently started clinical trials on this. And that would just be so cool if Foldit helped get rid of celiac disease. I'd be very excited about that. And many of my friends, gluten intolerant, would also be as well. In 2014, UC Davis students won the iGEM competition by using Foldit to create a biosensor that detects rancid olive oil. I didn't know this, but a lot of the olive oil in supermarkets is apparently, in grocery stores, is apparently rancid. That's super gross. Sorry about that. So anyway, they created a biosensor using Foldit. Foldit is also a great tool for education. It's been used in textbooks. Educators assign Foldit puzzles as homework, kind of get to the fun part of science first. And before the pandemic, we were working on a Foldit education mode, specifically for undergraduate and graduate education. But once COVID hit, we tried to release it as quickly as possible before fall of 2020. And we're now constantly updating it as we get feedback from educators. And if you're an educator, you can check it out at fold.it/educator. And yes, our website is fold.it. We're also very proud of that. I found Foldit to be super useful for getting kids excited about science, whether it's at local science museums, at science fairs, or local schools. We volunteered at the free USA Science and Engineering Festival, which is the largest science festival in the country that has unfortunately been on hiatus due to COVID since 2018. But we presented Foldit to the community there, and multiple times I had a wonderful time. And the NIH, National Institutes of Health, even had a table about the different games in science that are out there. Obviously with Foldit, but I kind of wanted to show you the other ones, which are really amazing as well. One of the co-creators of Foldit at Carnegie Mellon University started an RNA folding game with colleagues at Stanford. It's called Eterna. And the cool thing about Eterna is that they're able to quickly solve many player solutions to then give them feedback very, very fast, which we cannot do with protein. Solving a protein, as I said, takes a long time, really expensive, and doesn't always work. But so that's one really neat aspect of Eterna. And plus you can make some fun shapes with 2D RNA. This is Philo, a multiple sequence alignment game created at McGill in Montreal, where citizen scientists take DNA sequences of different species, so you can see here, and try to move them around so that they match as well as possible. And you can see here, the game does not show you any DNA letters or anything, just different colors representing the 4 different letters that make up DNA. Now, have you heard of a video game called Borderlands? They took Philo and they put it in Borderlands 3. In a new mini game they call Borderlands Science. And yes, that is poo at the bottom. Borderlands Science produced 5 times more data in the first 12 hours than Philo did during its 10 years of operations. That is a lot of data. Another game is iWire, which was developed at MIT to map the 3D structures of neurons, which is a very difficult 3-dimensional task. Another thing that, again, humans are particularly good at, as opposed to computational methods. Stall Catchers is a game trying to fight Alzheimer's. Very quick story about that game. I teach a gamification class at UMass Dartmouth, and I brought up Stall Catchers to my student as homework, and I don't know why, but I said whoever gets the top score gets extra credit points. And I warned the Stall Catchers people about this because I'd done it for a different program called Malaria Spot, and my students had hacked it, and yeah, I felt bad about that. So the Stall Catchers people, however, said, "Bring it on. We would like to, make sure ours is secure. So, do your worst." Anyway, I posted it at, I think it was 10 a.m. on Sunday, and I woke up, checked my email Monday morning, and there was an email from the founder of Stall Catchers going, "Okay, please stop. You win. Just hold on." And yeah, so I figured, as hackers, you guys would like that. So these are all different examples of how computer scientists can team up with other scientists to tackle and try to solve important problems in the life sciences. Has anybody ever heard of the game EVE Online? They even tackled COVID-19 as well. One of the games that EVE Online used in their mini games had actually started as a mobile game, Colony B. The mobile game was created in collaboration with the American Gut Project, studying the microbiome. It's the same team that works on Philo and Borderlands 3, which is why there was poop in their emojis. This was the first mobile citizen science game I found. But in fact, a lot of families would ask me at these science and engineering festivals, why can't I play Foldit on my smartphone? And it goes back to how much computation is required for the protein folding problem. Your phone battery would die in a couple minutes. So, what would be a possible solution to this? Well, one obvious one is just put all the computations in the cloud, which is great, except that somebody has to pay for it. Our game is free, and as academics, we don't have that much money. So Foldit remote control is kind of what we came up with, where all the calculations are done on your home computer, and then you can control everything remotely on your tablet, like this. So these are the important calculations that would melt your tablet really quickly. You can also do multi-touch, where you can grab different sections at the same time, like that. And if you combine these two, you can do multi-touch and remote control. But it's more fun to play with your friends rather than with two hands in the same place. Maybe one day we can all play Foldit in our living rooms with the whole family. This was back in the Connect days. But more recently, we've obviously wanted to integrate Foldit with VR, well, for many years now. But we haven't been able to secure any funding to do so. And that's kind of an expensive task. That just looks like Street Fighter at this point to me. But anyway, so that's Foldit. If you want to check it out, again, fold.it. It is the epitome of a massively collaborative project between many different institutions, and most importantly, collaborating with citizen scientists all around the world. So I want to always thank them last, because without the Foldit players, there wouldn't be anything like this. Thank you so much, and take care.